Hello, today Malaysia celebrates our 64th Merdeka Day. On this most auspicious day, we look at the everyday Malaysian that has achieved so much and has accomplished much more to inspire the many of us to follow in their giant footsteps. One such outstanding Malaysian is none other than Dr. Serena Nik-Zainal. Based in the United Kingdom, Dr. Serena has just been awarded the Francis Crick Medal and Lecture 2022 in recognition of her contributions to the current understanding of the causes of cancer. The award was announced by the United Kingdom's Royal Society just last week, and her work into the analysis of permutations and mutations present in cancer cells are now being applied to cancer treatments. Dr. Serena is a clinician and a scientist based at Cambridge University's Centre Research MRC or Medical Research Centre unit. And there she leads a research group that looks at computational biology techniques that studies mutations found in cancer cells. But her outstanding work did not actually begin here. In fact, Serena and her team had previously won the Dr. Joseph Steiner Award in 2019 for their research on mutations in cancer tumors, among others. Dr. Serena joins us from Cambridge, United Kingdom. How are you, Doctor?
Thank you so much. I'm good, thank you. How are you?
So it's like this. I was talking to my producers before we had this interview, and we're like, okay, it's not that often people can say that they cured cancer, but you quite literally have. So how does that make you feel?
Oh, that's very, very kind and a very generous introduction. So no, I think I better quickly put that a little bit more correctly and say we haven't cured cancer. I haven't, and certainly many others haven't either. But we have been able to make some inroads into using some of the genetic information that's present in cancers and to try to understand cancers in a whole lot more depth given those mutations.
Right. So the first things first, you were a clinician, and of course now you're a researcher. Let's try to understand the move towards being a clinician to a researcher and now holding both hats. What motivated you to do more work in cancer research? What motivated you to do genomics medicine and bioinformatics at that?
Okay, so I came to the UK very fortunately on a Petronas scholarship to study medicine, and that was in Cambridge. And I really love doing medicine. I still do medicine. I still do about 10% of my time spent in clinical work. But I'm a clinical geneticist, so I deal with families that have rare genetic disorders. And during my time training as a clinical geneticist, new technologies became available. So this is work by other people. And they were able to read the human DNA, which is enormous. The human DNA is 3,000 million base pairs or building blocks. But they were able to create a technology that was able to read all the DNA building blocks very, very quickly. And so this is called next generation sequencing technology. And when that was coming to the fore, I decided that I wanted to learn a bit more about it because I knew it was going to come to us in the clinic. I wanted to be able to understand it. I felt that I had to understand it if I was going to be able to talk to my patients about the genetic conditions that they had. And so, I ended up applying to the Wellcome Sanger Institute, which is one of the human genome centers in the UK that was involved in sequencing the whole human genome. They were one of many, many centers in the world that together tried to understand every single one of those 3,000 million building blocks. And because they created this map, let's call it a map of the human genome, today people like me can look at cancers and we can compare a cancer's genome to that map of the human genome and show that actually every person has got loads and loads of mutations in their cancers. And so we then showed that when you study the mutations in human cancer, you can actually learn a lot about the causes of everybody's cancers. And everyone's cancer is so unique, it's so individual. And so, my team were very, very lucky. I think we got into the field just as next generation sequencing was taking off. And we learned how to use all the mutations in the cancer genome to basically report what's wrong with every single person's genome in a very detailed way. And that's how I've landed up where I am today, It was not my plan to be a scientist necessarily. I was I wanted to learn more about it, and that's how I ended up in cancer genomics and doing bioinformatics. But I've loved it, and I've met so many people who are passionate about it and who want to try to bring it to clinic. And really, that's what's driven my team and I to keep pursuing this path and to keep creating ways of trying to read the cancer genome and make it easy and interpretable for people to use, for patients.
So right now you are at the forefront of cancer research. Your next generation sequencing idea is of course massively parallel towards what is required to push forward the boundaries of medicine because of the ability for it to offer high throughput, scalability and speed. The technology of course is to determine the order of the nucleotides in the entire genomes and targeted regions of DNA and RNA. That requires you to be able to sieve through this data quickly and correctly. So there's a lot of artificial intelligence being done on this. I don't know. Are you also taking up, coding, computer technology knowledge in order for you to make sure that the work that you do in next generation sequencing is going to be done well and accurate? How do you marry these new knowledge with the kind of technology that is needed to get it done? Or do you have to rely heavily on some team members that would be quite proficient in some areas and you provide value in some other areas?
Right. So this is a great question, and I think you've basically alluded to it already. The amount of data that is generated from the mutations in cancers is a huge amount. So there's no way that I can manage this by just using an Excel spreadsheet or PowerPoint. We have to use computational methods. So you're absolutely right. During my PhD, I had to learn how to code. I had to learn how to use computational methods to handle that data. And so I have some skills, bioinformatics skills. We need to have some statistical skills as well, and analytical skills. I mean, a lot of it is sort of standard scientific methods, but just used in a computational way. And now my team comprises about 70% computational people and about 30% wet lab. So we do also have a laboratory where people are doing experiments. And there is, in today's world where there's just so much data and so much information, you can't really manage with sort of one silo of working or styles of working. And so we in our team have a mix of people, biologists, we have statistical people, we have purely computational people. We also have people who have skills at making websites and interfaces so that people can use IT things easily. So there's a spectrum of people in my team. The clinical people are also extremely important because, of course, they see the patients and they know what the questions are. So really having this sort of multidisciplinary sort of dependencies, I think, is extremely important. And one of the reasons why I think we've made so much progress so quickly.
All right. Of course, when you mentioned that split between 70-30 kind of makeup, it's clear that the 70% is going to be driving a lot of the legwork in terms of trying to make it work, the complex genomic questions that require a depth of information beyond what you guys know right now, beyond the capacity of traditional DNA sequencing, for instance, and because of the different permutations that cancer research brings about, you guys have to be very dynamic. My question to you is this: how often do you have to rely on human intervention versus, relying a lot more on AI to do the work? Do you feel that human intervention is very much critical at this point in time in your research?
Absolutely. At the end of the day, the computational methods, the algorithms that we create, whether it's, we use a lot of machine learning, so it's a subset of AI. It still requires human intervention. It still requires a person to say, what is the question? What is the thing that we're trying to achieve here? So, for example, if you have a whole lot of colorectal cancers, so cancers of the colon, and you want to be able to classify those cancers into the ones that could be sensitive to one drug versus another, you still need human input to try to say which ones are in one group versus another, and then you teach the algorithms, the computational algorithms, to learn the predictive properties, and then you use those algorithms on new patient data sets. So today, we hear a lot about artificial intelligence and machine learning methods in computational healthcare, But actually, we're still very early on. People are still learning and requiring a lot of human input into trying to ask the right questions, to design the right algorithms, to design computational methods. And then, there's another very big step, which is trying to show that it's useful, because you can create algorithms, but to show that it's useful, you've got to demonstrate that it can improve a clinical outcome. So does it improve your likelihood of being sensitive to a drug? Does it improve your overall survival? And those require clinical trials. So, computational methods are very powerful and are fantastic, but they still require those clinical trials, the rigor of doing evidence-based medicine before we can really put all of them into clinic.
You straddle between two very diametrically oppositional roles, if I were to say so myself. Because being a clinician, you deal with human beings, you deal with emotions, you deal with families. And in the realm of oncology, you're dealing with patients that might not necessarily have the best odds to survive, unfortunately, but that is the case. But at the same time, you are also a scientist. You are going to be driven by the weird and the fascinating realm of science, and it doesn't stop, because scientists have that thing where the more you do your research, the more you realize you don't know, and the deeper you dig into this rabbit hole, and you ignore everything else, including that human aspect that I just spoke of earlier. How do you ensure that what you do in the lab is going to be translated into how you communicate, not just to the patients and their family members, but to the folks like me, a person that is, might not be necessarily to be too familiar about the realm of medicine, of genetics, of computer? How do you reach out and manage these very different roles that you have right now?
That's a very perceptive question. So, and actually something some of my colleagues need to sort of think about. But I think it's a very, very good question. So I think it's two things. So the first one is that, or maybe three things. The first one is that I still do my clinic. So I still see patients in clinic, and I enjoy that. It's very different to my research. You are absolutely right. And my clinic, remember, I'm a clinical geneticist. I'm not an oncologist, so I don't treat patients with cancer. I treat or I manage families that have inherited genetic disorders. So they'll have children with learning disabilities or have some congenital abnormality that you cannot get rid of, or have a family history of cancer or neurodegenerative disease. So it tends to almost always be bad news. And it's very humbling to do those clinics. And I always remember that, life deals you a hand, and you don't really get to choose. This is your genetic cards, or this is your life situation, or these are your parents. This is just the situation that you were given, and these families have to sort of deal with it. And so I do find it very humbling doing that clinic and doing those clinics. So I still do them. And I don't do anything special. I don't do like my own specialist clinic or anything. I do a general clinical genetics clinic. I think it helps me keep my feet on the ground. The second thing is family. So, my mom, unfortunately, she has passed away now, but she was always one of these ones who was, very quick to sort of, even if we had a major publication or a prize or anything, she would say, so how does this benefit mankind, right? So, how is it useful? Because to her, the publications didn't really matter. We as scientists, people are sort of busy notching up their prizes and their publications, but my mom was very sort of, worldly and would sort of, make sure that I stayed grounded. And I think the third thing, I think a very important thing, is to stay involved with patients when you do your research. And so increasingly, we have something called patient-public involvement, PPI, and patients and the public actually get involved in the science. So they are involved in when you are writing a grant. They're involved in when you're writing a paper. They're involved in communicating your message out to the public. And, some funders, some of the people who provide us money to do research have sort of said, you have to do this. And I think some people think of it as a box-ticking exercise. But I've found it a very illuminating way to ensure that your work stays very human. And actually, some of the patients and the public that get involved, because they have lived experiences, they've been through this in their lives, or they have looked after patients who have died with these different diseases, they can bring a perspective which is human, which is down to earth and grounded, and it also reminds you why you are doing it. You know, that you're doing science for the benefit of society and mankind, as opposed to doing science for your personal satisfaction. So I think those are kind of the three main ways I sort of try to keep the balance.
Yeah, that's right. Because there's no point doing research, and I mean, people keep on talking about this, the mad scientist syndrome, because if it's not actionable, it's not translatable into why you're doing it. That why is very important. We'll go for one short break. When we come back, we'll discuss a little bit more with Dr. Serena of Cambridge University. Welcome back. We have Dr. Serena Nik-Zainal joining us from Cambridge University. She's a university reader in Genomic Medicine and Bioinformatics at Cambridge. Dr. Serena, before we jump into all the other stuff, I just want to ask you whether or not this Merdeka Day feels a little bit special. It's not that often that somebody wins the Francis Crick Medal. What would be some of the messages that you want to share with the many Malaysians right now that are watching our conversation today?
Thanks so much for the question. It's bittersweet, actually. It's of course it's delightful to be able to win this prize. I'm a little bit sad because my parents are not here to sort of witness it. I'm a bit far away from family. It's the pandemic. There's a lot of things to be a little bit sort of sad of. But I'm very grateful for the honor and the recognition. And I do feel quite strongly that what the pandemic has shown us is that one of the areas that really is growing and will continue to grow is data science. So this is big data. So when you have loads and loads of information, whether it's mutations, whether it's clinical information from COVID, whether it's public health information, big data is on the rise, and it's here to stay for every country in the world. Not just in the UK. It's also in many other places in the world as well. So I think for the next sort of generation, as it were, and for planning and anticipation, I would urge the next generation, if you're at all interested in science and technology, to please think about learning how to code, get an understanding of statistics. If you are interested in science, technology, and medicine, it's just a really useful tool. And if it has become so huge in the few years that I have been in science, I think it's going to dominate your lives in the future. As for our leaders in Malaysia, I would say look at the infrastructure that is available. Is it adequately future-proofed? Are we equipped in terms of investment into hardware and software? And are we upskilling people to ensure that they can stay abreast with developments nationally and internationally? We basically need to prepare our next generation and have the right infrastructure so that they can keep up with the developments and the advancements that are coming. Those are very lovely messages, actually, to be shared. My own daughter aspires to be both a scientist and a medical doctor, but she looks at her mom and she's really spooked out by the life of an HOMO specialist and now consultant. She's like, she might not go through this path. She said, I want to be like you, father, and just sit in front of the TV and chat with people. I think that's a lot more fun. And she encapsulates that particular argument here. There's a lot more fun things to do, and then there's a lot more real hard things to do. And I'm not trying to diminish my role here, but I'm also trying to underline the fact that you guys are actually doing the dirty work here. You guys are doing the heavy lifting. And this has brought about that whole concept during the pandemic that a lot of people are now arguing, that there are still a lot of people around the globe that might be suspicious about science, might be skeptical about the findings, might be hesitant or outright oppositional towards medicine and research.
there seems to be a lot more outreach and communication that needs to be done. When I say, you know, a lot, there's a lot, right? Across countries, across socioeconomic structure, across first world, third world, across all that, across religious, non-religious, secular countries, there are sections of people that is still oppositional towards the work that all scientists and doctors and medical professionals and all the other people that like you do. How do you intend to push forward and make your work not just relevant but communicatable to these individuals, to this sector of the society that needs to be getting along with the progress, the train of progress, in order for humanity to chug along these very trying times? How do you make sure that your work is not going to be diminished by these folk? That's my big question to you right now.
And I think it's a very big question to try to deal with because, of course, as we've seen in the pandemic, that misinformation or disinformation has been extremely destructive. So I think there's a humongous piece of work that needs to be done on science communication, on medical communication, and to get it out there sooner, to get it out there as quickly as possible, but in terms that can be understood by people in ways that they can value. And so I think, and of course, you know, science communication has become a really big field. I mean, there are people who are, you know, professionals in scientific communication, and I certainly am not one of them. But you know, I think a lot of scientists and medics.
I don't know. I think you're communicating pretty well. I mean, I kind of understand 90% of what you're trying to say. 10%. Okay, let's not overinflate the whole thing. But yeah, I don't know. I feel that the realm of communication has to be shouldered by everyone, including people like yourselves. No?
Yeah, I agree. I totally agree. So I think that is exactly right. And I think, you know, I do operate in, you know, a sphere of Twitter where I can see how we lose contact with the general population because we're so absorbed in, almost a little bit absorbed in ourselves. You know, we sort of start to discuss certain very bitty things, and then we lose the bigger picture. So you're right. I think everyone has a responsibility of ensuring that, you know, for your work to stay relevant, you need to be able to communicate it, and you need to be able to communicate it to a layperson. And in fact, when you apply for a grant, for a scientific grant, it is a requirement that you can summarize your work. Unfortunately, in only one part of nearly every grant, one little paragraph to a layperson, and usually it's so high level that you can't really capture it. And what I think we fail to capture from scientists and doctors is the outcomes, right? They write the layperson summary at the start of the grant, but they never ever summarize it at the end. And I think that's the bit that we need to do better. Because if we can put it into simple words and be able to explain to school children why something is important, on their terms, why it's important for them in the bigger picture, then, you know, I think we'll go some way towards trying to reduce the amount of misinformation and disinformation there is out there.
Right. Before we end, I just want to ask you what's next for you. Is it more cancer research? Is it something interesting else? Something that, you know, you're going into Silicon Valley and try to build AI solutions for medicine? What's next?
This is a great question. You know, some people sort of plan things for 5 or 10 years in advance, and I never do that. I always sort of go, what keeps me awake at night, and what am I doing now?
I find that hard to believe. You're a scientist. You got to have some form of planning, right? I mean,
when you write your grant, you have to say stuff for 5 years. But you know, as easily as things come, things can go, and as quickly as something can be, you know, flavor of the month and can be all hyped up, it can very quickly go as well. So I think, you know, you just have to be a little bit smart about it. And so my team are driven by, you know, we want to make our stuff useful. So we want to put it into the healthcare system. And some of these machine learning AI tools that we have built, we want to put into the medical health system in the UK. But then my next thing is I want to make it available and amenable to beyond the UK as well. And, you know, I would love to see it sort of, you know, get embedded into the healthcare system in Malaysia, for example, and in any other country for that matter. So for us, we want to get it into the healthcare system. We're also looking at other areas, not just cancer, because, in fact, as we age, all our cells are changing. The DNA in all our cells are changing as we age. And so we're also looking at some other diseases, not just cancer, so neurodegeneration, and trying to understand how, the cell changes and actually how the cell manages to survive in spite of all the things that we do to our cells. So, so we're exploring some other areas, largely to do with aging. I'm quite interested in trying to understand how we age healthily going forward. But I think one of the biggest drivers for us is to make our things as useful as possible to as many people as possible as well.
Thank you. All the best. Selamat Hari Merdeka to you, Doctor. That was Dr. Serena Nik-Zainal joining us from Cambridge, United Kingdom. She is a university reader in Genomic Medicine and Bioinformatics, Cambridge. Dr. Serena has just been awarded the Francis Crick Medal and Lecture 2022 in recognition of her contributions to the current understanding of the causes of cancer. That's it for now. Happy Merdeka, everyone.